Yeah, this was a really great science that came out of Robbie Meisner's lab and group at Stanford University. Again, I thought it was one of the best, if not the best, ASH abstract this year. So they analyzed the tumor DNA either from the blood or from actual tumor biopsies in patients who had received axicabtagene ciloleucel, and then they tried to look at the long-term outcome and the responses of those patients. And what they've identified was that there was a loss of CD58, either mutations in or actual loss of the molecule in CD58 that was correlated with either lack of response or early relapse in patients, again, treated with axi-cel. And so what they were really, what they showed was that CD58 essentially interacts with CD2, and CD2 is providing a stimulatory signal to the CAR T cell. And if the CD58 is lost, then you don't get that CD2 signal. And essentially it seems like that's not enough to trigger the T cell to be able to eliminate all of the tumor cells. And so then they've actually proposed a dual CAR mechanism where they, instead of having like 4-1BB, a co-stimulatory domain, they actually link on the stimulatory domain of CD2 and use a dual CAR, and then maybe be able to avoid this CD58 loss or mutation. And so I think it's just an outstanding analysis of patients treated with a commercial CAR and being able to then dive deep into why do some people lose their early response and then relapse, and then they actually have proposed a way around that. So we'll have to see how those trials work in the future.